Alright, but that's not the end of the cycle, of the Calvin cycle. The leftover G3P, and there is going to be some leftover G3P, is going into the last phase. And the last phase of the Calvin cycle is regeneration of RUBP, which pretty much means it's going to take these leftover G3P molecules, it's going to convert them back into RUBP, so the Calvin cycle can use the leftovers again. So it's going to make, and what RUBP stands for, it's going to stand for this: ribulose type of sugar biphosphate. Alright, that's our RUBP. And then the RUBP is going to be used again. Okay, and the cycle continues. So that is in essence the Calvin cycle. It takes in carbon dioxide, combines it with RUBP through carbon fixation, so it can turn this carbon from carbon dioxide into a usable form. This new molecule that's made as a result, this PGA, is going to, energy from NADPH and ATP is going to be added to it in the reduction part of the Calvin cycle to make G3P, this glyceraldehyde 3-phosphate, which is very important because that is going to combine, is what's going to be used to make the sugar, and the sugar is the end product. Alright, so that is photosynthesis. We have where it happens. We know what it happens in: plants, certain green algae, and then certain types of bacteria. Now there are factors that can affect, let's go and zoom in here, there are factors that can affect photosynthesis, such as the intensity of the light. So morning sunlight versus noon sunlight versus evening sunlight, that's going to be different amounts of intensity. Okay, cloudy versus non-cloudy. The amount of carbon dioxide. So CO2 concentration. Okay. And then temperature. A certain range works best. So if it's too hot or too cold, it won't work as well. Because actually if it's too hot, the stomata are going to close and it won't let, to keep from losing water through evaporation, so it won't let carbon dioxide in. If it's too cold, that's going to affect these enzymes that are involved in the process. And this is your photosynthesis.